Okay, so this is Dawn Van Dam. I'm the General Manager of Cambridge Health Tech Associates, and I would just like to welcome everyone to today's webinar. I'll give a brief overview of the topic that we're going to be covering, and then I'll introduce the speakers and turn it over to them. So today, this webinar is called Metagenomics, Microbiomes, and Models, oh my. This webinar will feature a discussion with leading experts in three areas. One, antibiotic resistance in human-associated and environmental bacteria. Two, omics data. And three, the NIH Human Microbiome Project. This effort to drive forward the science of metagenomics will feature presentations by three of our speakers, which I'll get to in a moment. And we're very excited because we'll be hearing information about multi-omic modeling of environmental and metagenomic insights from the University of Chicago's built environment work. Also, we'll focus on antimicrobial resistance and also the NIH project looking at an overview and some of its major products. So just to tell you a little bit about us, today's webinar is being sponsored by both NGS Leaders and Cambridge Health Tech Associates. NGS Leaders is a free online community where we host discussion forums, blogs, and webinars. So we would encourage you to join and to become an active member and to fully participate. NGS Leaders is organized by Cambridge Health Tech Associates, and we help out in the area of science by helping transforming science into commercial success. And we do that three ways. So through consulting and market research, through technology evaluations with the pharmaceutical and biotech industry, and then through marketing and communications. So there's just an overview of our three different areas of service, and if you would like to have us help you transform your science into commercial success, we'd love to talk to you about how we can do that. As you probably know, we did expand our consulting and research practice not too long ago in the area of next-generation sequencing, and one of our key people involved in that is Dr. Kevin Davis, who has been known in this area for over 20 years. So moving on to today's participants, Dr. Jack Gilbert, Dr. Gautam Dantas, and Dr. Lisa Proctor. I will give a brief overview of everyone's background, and then we will jump into the presentation. So starting with Dr. Jack Gilbert, he earned his PhD from Nottingham University in 2002 and received his postdoctoral training in Canada at Queen's University. He subsequently returned to the UK and worked for Plymouth Marine Laboratory as a senior scientist until his move to Argonne National Laboratory and the University of Chicago in 2010. Dr. Gilbert is an environmental microbiologist at Argonne, Associate Professor in the Department of Ecology and Evolution at University of Chicago, and Senior Fellow of the Institute of Genomic and Systems Biology. Dr. Gautam Dantas is an Assistant Professor in the Department of Pathology and Immunology, the Department of Biomedical Engineering, and the Center for Genome Sciences and Systems Biology at Washington University School of Medicine. His research interests and training lie at the interface of microbial genomics, synthetic biology, and computational biology. He is the recipient of the AAAS Newcomb Cleveland Prize, the Harvard University Certificate for Distinction in Teaching, the NIH Director's New Innovator Award, and the Kenneth Rainin Foundation Innovator Award. And then Dr. Lisa Proctor, as Program Director, is responsible for coordination of the Human Microbiome Project, or HMP. The HMP is an 8-year, $194 million trans-NIH Common Fund initiative to create a community resource of data, research resources, and clinical and scientific approaches for this emerging field. Dr. Proctor joined the NHGRI Division of Extramural Research in 2010. Prior to this, she served as Program Director at the National Science Foundation in the Geosciences and the Biosciences Directorates, where she managed microbiological and bioinformatics research programs. So with no further ado, I'm going to turn it over to our first speaker of the day. That would be Jack Gilbert, and he's going to take it away and give you his presentation today.
Okay. Thank you very much, Dawn. Appreciate the time. Can everyone hear me? Well, I'm assuming I'm audible now. Okay, so I'm going to be talking today about some of the work we've been doing with environmental modeling and mapping of microbial taxonomic and functional potential across different ecosystems. And I have a short period of time, so I'm focusing this presentation on work we are doing inside the Earth Microbiome project and also microbialmodels.com, which you can see there on the front page. If you're interested in the built environment research, then we also can you can go and see those on home microbiome and hospitalmicrobiome.com websites, and our public-funded human gut and oral cavity initiative, AmericanGut.org, is also visible there on the front page. So I'm very interested in the microbiome of different ecosystems and how they respond and change in regards to different capacities that we have. About I don't know, 3 years ago, myself and two other slightly crazy individuals, Rob Knight from the University of Colorado and Janet Jansen from Lawrence Berkeley National Laboratory, came up with a new research initiative called the Earth Microbiome Project. The inference for this initiative was to, in the first instance, create a systematic characterization of global microbial diversity. And we predicated this on the potential that we didn't have enough money, or no one would give us enough money to actually go out and examine all of the environments we were interested in by ourselves. You know, Rob Knight's very interested in animals and human-associated microbiota, and Janet's interested in soils and permafrost and those kind of things, and I'm very interested in marine and fresh water and lakes and rivers, etc. So together we kind of said, you know, we span some of the environmental microbiological work. Here is something we might be able to do: bring in our colleagues who have already collected samples, who would be willing to share those samples, those physical samples with us, and then we would take those samples, extract them, PCR them, make metagenomic libraries and sequence them and analyze them in a coordinated and comparable fashion, so that you could compare across all of these different ecosystems and generate this systematic network of understanding for microbial diversity. Primarily, we're interested in exploring the patterns of microbial diversity across different ecosystems. By taking all of the metadata, the physical, chemical, and biological variables for each environment that we have, we were interested in how those processes, those physical, chemical, and biological parameters, drove patterns of diversity. And then we're also interested in how the systems and scales associated across time and across space, and that comes to the modeling of that data set. Obviously, this is a crowdsourced effort, so we work with the community to gain access to samples and their physical and chemical and biological metadata, so we can process and analyze that data set consistently. And it creates a framework for generating predictive understanding of what environments may provide or may generate which kinds of microbial communities, so understanding niche modeling and niche variability within ecosystems. So we can determine the emergent properties of microbial dynamics. It's being developed for the community, by the community, with immediate, free and open access data to provide a comparable environment in which any researcher, even if they don't want to donate their samples to the Earth Microbiome Project, they can generate their own data. They can use our standard techniques, which are freely available on earthmicrobiome.org, and generate their data and put it in the context of this global effort. So if the numbers, we currently have more than 100 collaborators on board who have donated or pledged around 60,000 samples. We're currently processing around 22,000 of those. Over 20,000 are being completed already for 16S ribosomal RNA analysis using the V4 region of the 16S gene. And we generated about 1.7 billion 16S fragments. And those are all available online. We've generated some metagenomic data across a number of different ecosystems, but that's very slow going. We can generate a lot of it, but not from many samples. Currently, from the first 10,000 or so samples, we've identified around 6 million bacterial OTUs, about 97% nucleotide identity. across that 16S-V4 region. So that's a huge amount of diversity and comprises the largest effort. In fact, the analysis of that, to get to that, was the largest operational taxonomic unit picking effort in history and burnt through a hell of a lot of Amazon computing, cloud computing time. And that public analysis is being led by Greg Caparasso and Sean Gibbons so Greg's at University of Northern Arizona and Argonne National Laboratory, and Sean's at Argonne National Laboratory and University of Chicago. We're using this kind of information to generate predictive understanding, and to do that we utilize a niche modeling infrastructure whereby environmental parameters define the niche. We use that combined with a correlation to 16S RNA understanding of the community structure to predict what communities live there, i.e. what taxa and how abundant they are. Then we use metagenomic data and predictive tools to determine the functional metabolism of that community at any given time and location. And then we use that functional metabolism to determine the influence that that community at that time could have upon the environmental parameters, i.e. a sudden increase in nitrogen fixes may be significantly increasing the amount of nitrogen content in that system. And so we can spin this model and make it go around and around and around ad infinitum to predict overall ecosystem structure. And we use predictive algorithms embedded in a Bayesian network. So what you're seeing here is a Bayesian artificial neural net where the nodes in colors are bacterial taxa that are whose abundance is predicted based upon changes in the environmental characteristics, which are up there in those gray diamonds. Things like temperature and phosphate concentration and chlorophyll concentration and ammonia. But importantly, and this was published last year, we explicitly look at interactions between, or correlative interactions between the taxa, so that we can get a better prediction of how their abundance changes in response to changing abundance of the other taxa. So some organisms in here are purely a product. The ability to predict their abundance is purely a product of the changing abundance of another taxon. Others are driven by the environment. Others are driven by the environment and other taxa. And that's quite important. So we use that information to predict the community structure. We also can use it to predict the community's ability to modulate different environmental characteristics. And we call this predictive relative metabolic turnover, or the hairball, where we take shotgun metagenomic data, we annotate those fragments of genomic information to known functions, and map that onto a metabolic model, a metabolic pathway map, as it were, to create a community metabolome, whereby the changing relative abundances of the annotated enzyme activities in the metagenomes can be used to predict if a metabolite is being either more consumed by the community or relatively more generated. So you can look at accumulation of metabolites or consumption of metabolites across time and space. I don't think that GIF is going to work because we're in a situation, but what you would normally see here is, this is a snapshot rather than an animated GIF, of the ability of the community to modulate carbon dioxide metabolism in the context of all of the other metabolites in the vicinity. So you see there right in the middle you have CO2, that's carbon dioxide obviously, and it's dark at that time. That means it's being relatively consumed by the community. And CO2 is more consumed in this marine system when you have autotrophic activity dominating. So you've got photosynthesis consuming carbon dioxide. If you saw this during the nighttime, that CO2 would be bright yellow, which would mean that it was being more generated by the community, and that would infer respiration. And we can see how that plays out. But essentially, we can correlate these predicted changes in carbon dioxide to observe changes in the actual partial pressure of carbon dioxide in this water column, for example. We get about an 85% correlation between those two observed measurements. And that's fantastic, because that means that the microbial dynamics of this system, the ability to predict their metabolic potential, is effective in our capacity to take metagenomic information and predict how they may influence ecosystem variables such as the partial pressure of CO2. Now, so if we encapsulate that, we can take that satellite image modeling data, and I didn't really talk about that, but that's the hyperspectral imagery that predicts what the characteristics of an oceanic environment, or indeed a terrestrial environment, might be. So you can predict things like temperature and the amount of chlorophyll and even the concentration of different chemicals. We take that satellite data and that predicts our niche. Once we have the niche, we use microbial assemblage prediction to predict the community structure across space and time. Normally this would be a moving GIF, but here you're seeing the different colors represent different Bray-Curtis community composition scores compared to the average, whereby the blue and the purple are communities that are very similar to the average. The green and the red are more dissimilar to the average. They are communities that are very unique compared to the average for that entire area. We predict that over nearly 6,000 49 kilometer square grid cells across the entire ocean. We can do this for about 4.5 years at the moment. And we can use that information with predicted relative metabolic turnover to predict the ability of that community to either consume or generate carbon dioxide. And of course, again, that's entirely based on the ability to synthesize the metagenome, so understand the changing relative abundances of genes in that community. And here you can see, the majority of the carbon dioxide potential at this time is either being more generated in green or slightly more consumed in purple. And there's no extremes across this English Channel basin, but the consumption of CO2 would infer an increase or a slight, favorable balance towards photosynthesis, and the generation would infer a more favorable balance towards respiration. I'll skip that bit. So skipping on to the Earth system model, so I've shown you that we can do this quite effectively for marine systems. Can we do this for Earth system, terrestrial systems? So we think we can, and no one can prove us wrong at the moment. But this is what you're seeing here in this rotating globe is soil moisture, the ability of the soil to in this system to we can predict the physicochemical parameters of the soil across this terrestrial environment for the Earth. And the Earth system model is a national and international effort led by companies like organizations like NOAA and the Department of Energy, whereby it enables us to generate predictions of things like soil moisture. But we can also generate predictions of 12 other different parameters, such as different concentrations of soil carbon. And so what you're seeing here is soil moisture across the globe for January 2005. Now these 12 parameters, including soil moisture, define our niche. So now we know what the niche of the global terrestrial system may be in January 2005 as an average. Using that information, we can predict using microbial assemblage prediction, this MAP algorithm, we can predict the microbial community abundance for different taxa. So I'm looking at a very high level phylum level taxon here. For alpha protobacteria is actually a class, the alpha class of the protobacteria. And what we see here is a differential relative abundance of alpha protobacteria across the globe as predicted based on the niche structure. Once we know what organisms are there and their relative abundance, we can use that information to predict their relative gene abundance based on correlative indices between the taxonomic abundance and the abundances of genes in the metagenome, and also the abundances of genes associated with comparable taxonomic genomes that have been sequenced in databases like GenBank and IMG. We use that information to predict the abundances of genes. So what you're seeing here is cellulobiosidase, the gene that codes for cellulobiosidase, and its relative abundance across the globe for January 2005. Once we know the gene abundances for all of the genes in the system that we can annotate, we can use that to predict the ability of the community to modulate carbon dioxide metabolism across the surface of the Earth. So the red and the blue refer to differential consumption and production. So I'll show you that now. My last slide here, some of the arrows are twisted for some reason, but my last slide here, what we're seeing is the capacity to take that information from January 2005 and extrapolate it up until January 2100. And unless you are a Time Lord with your own with your own time machine, you can't yet prove us wrong. But essentially what we're doing here is taking climate models and using them to predict the niche structure that microbes may live in across the terrestrial surface in January 2100. Here's two different scenarios. So on the top we have a reduction in carbon dioxide emissions, and on the bottom we have a business as usual strategy. And what we can do over here with the, if you see on the right hand side with the black and yellow globe, we can look at the differences between those two climate forecasts, whereby yellow represents a relative increase in the capacity of the bacteria to consume carbon dioxide in this system. And carbon dioxide in this system is obviously linked to plants in many cases. So it's the respiration capacity of that community and the need of the bacteria for carbon in the form of carbon dioxide to fix and consume heterotrophically the respiration content of that community. So in essence, that enables us to drive forward the community that we have in that system. Then we go on to Gautam's presentation. So thank you very much.
Great. Well, thank you, Jack. That was fabulous. And while we're switching to Gautam's presentation, I just want to remind everyone, this is Dawn Van Dam, the General Manager. And if you can just choose my name in the chat box and send me any questions that you'd like to ask the speakers, we'll be doing that at the end of the 3 presentations. Okay, so over to you, Gautam.
Great. Hopefully I'm unmuted at this point. So yes, thanks again for giving us this opportunity to tell you a little bit about what we do on the metagenomics side. It's great to follow Jack, who's obviously doing some wonderful work. And so much like Jack, I'll only be focusing on a couple vignettes that we work on. I'll try to give you some introduction for the specific area that we care about, antibiotic resistance, and then go on to how we've developed and applied some of these functional metagenomic methods for looking at microbial communities. First off, though, I just want to acknowledge the folks in my group right now, and the reason I put them up, in addition to the fact that they do all the work at the bench, is that they draw from a variety of different fields, and this is something that we in the metagenomics community really are beginning to appreciate, that this really does bring across folks with multiple different interests and disciplines. And so, for instance, we have graduate students working on these problems across genetics and genomics, computational biology, ecology and evolution, and microbiology, and then also drawing postdoctoral fellows across pediatrics and pathology. So with that, the specific area that I'll discuss today in terms of how we've applied metagenomics is towards this spectrum of antibiotic resistance or antimicrobial resistance in pathogens. And so one thing we realize is that the treatment of antibiotic-resistant bacterial infections is an increasing challenge, and that's really an understatement. And so, just a couple of data points. This is not our work; this is culled from the literature. So, we recognize that globally, antibiotic-resistant infections are a leading cause of death. This is just some data from the CDC that shows just a single drug-resistant organism, methicillin-resistant Staph aureus, in the early 2000s, killed more people in the U.S. than did HIV/AIDS. Unfortunately, in the situation of a double whammy, MRSA being one example of many of these drug-resistant infections, but there are many others. So it's shown in this plot. You can see vancomycin-resistant enterococci, these fluoroquinolone-resistant Pseudomonas. All of those are going up over time, and unfortunately, at exactly the same time, the number of new drugs coming to market, the new antibiotics coming to market, has been sharply declining. And so you've got the increasing specter of resistance and the decreasing availability to treat them with new drugs. And so what that ends up meaning is a substantial amount of healthcare and human cost. And so, again, as estimated by the CDC recently, treatment of just the antibiotic-resistant infections in the U.S. incurs in excess of $35 billion of additional cost, translated from about 8 million additional hospital days. So that's the reason we care about this problem. Now, the central question then is, as these pathogens are becoming antibiotic-resistant, how are they doing it? And one thing that's clear from decades of work is that bacteria have the ability to exchange genotypes and hence phenotypes between fairly unrelated organisms. And so there are these traditional mechanisms altogether called horizontal or lateral gene transfer. The three major ones: conjugation, which is direct transfer of DNA between bacteria; transformation, which is the ability of some bacteria to suck up naked DNA; and then also phage-mediated transfer. But the main point that we and many others have recognized is that this horizontal gene transfer can occur not only between pathogens, but also essentially between any two microbes in any two communities, or multiple communities, for instance. And so depicted here are just a couple of examples. So the bugs associated with the human body, this commensal microbiota, bacteria associated with the soil, bacteria associated with animals, and in fact, there's probably a fairly complicated network diagram that depicts each one of these interactions. And today, I'll just describe a couple of these habitats that we've studied using metagenomic methods to look at the flow of antibiotic resistance. But first off, I want to say, why do we want to use these metagenomic methods? And so the traditional way in which you would study antibiotic resistance in the clinic is under this paradigm of Koch's postulates, where you try to isolate the specific exenic culture of a single organism that's the etiological agent, usually by plating things out on a petri dish. And then once you have that individual organism, then you can do all sorts of things. You can do a susceptibility profile. In this era of genomics, you can sequence the whole genome. Unfortunately, most habitats, as Jack has mentioned, are rife with a large diversity of microorganisms, most of which are not really amenable to traditional culture-based interrogations. And so for that reason, we use this alternative method called functional metagenomics to interrogate this reservoir of resistance genes. And this schematic shows how this generally works. We treat the entire microbial community as a grab bag of genes, and so that's the metagenomic aspect. We then extract all of this DNA at the same time, so this is now preserving the community structure in terms of what genes are there at relative abundances. We shear that DNA down into fragments that represent a couple of bacterial genes, clone them into some sort of expression system. So, for instance, a plasmid. This is the way traditional recombinant DNA work would progress, and then take this library of randomly cloned in fragments and pop them into some sort of easy-to-use genetically tractable host, in this case, E. coli. So this is the metagenomic library part of functional metagenomics, and the functional aspect is the next step, which is essentially taking this whole library of transformant clones and subjecting them to survival selection under antibiotic concentrations that would kill the wild-type host, such that now the only colonies that you see here are E. coli transformants that have picked up some random chunk of DNA from the original source, which is clearly encoding antibiotic resistance because it went through this functional bottleneck. And then, of course, the devil is in the details, where you go through, and this is where we spent a lot of time, to specifically analyze the sequence of these cloned out fragments after they've gone through this functional bottleneck by using next-generation sequencing methods and some multiplexing, and then a fair amount of computational work to try to assemble those fragments back up and get as high resolution of prediction of the gene annotation as possible. And so this is described in detail in a fair number of publications. And so the first vignette then is to apply this method to try to understand the role of the commensal microbiota, this group of important symbionts of our human bodies, as what potential role they might play in the exchange of resistance with pathogens. The reason we believe this is important is because we know over the last many years, over the last century basically, antibiotics have been dosed in the human population at a pretty high clip, and so that should give all of the selection pressure required for these commensals to become resistant. And then we also know that we're outnumbered 10 to 1 by microbes in the human body in terms of human cells, and hence just by a numbers game, a pathogen coursing through the body is actually probably bumping into more microbial cells than humans cells, and hence this is an opportunity for exchange. And so we're now applying this high throughput functional metagenomic method to look at things like the diversity, dynamics, and evolution of resistance genes in human commensals. And so in a study that we published a few years ago, we tried to do a direct comparison actually of the resistance genes that you might see from two unrelated adults in their fecal samples. And basically the comparison was the genes that would be in the aerobic cultured bacteria from their guts, and so this is what you would traditionally study by culturing, versus looking at the same sources, the same fecal samples that these aerobic isolates came from, except now looking at the entire metagenome in this functional manner. And so what's depicted here in these two plots are the average sequence identities of these functional genes that came out to either any gene in the database in white or specifically resistance genes and pathogens. And so what you'll see is that these two plots look very, very different. Most of the genes that came out of this aerobic culturing look very similar to things that have been seen before, and indeed have high identity, in some cases 100% identity genes to pathogens, despite the fact that these bugs are from healthy humans. Whereas the genes that came out of the metagenomic sampling were largely novel, so with only 50 to 60% average identity to any gene in any database at the time. And so the major conclusion from this was that resistance in this commensal microbiota was severely undersampled because of this prior over-reliance on culture-based techniques. And so with that as a framework, we in our lab has focused pretty specifically in the pediatric gut microbiota, specifically of healthy kids. And the reason for this is that a lot of people have now shown that the microbiota is very dynamic during the first 3 years of life. And then after that, most humans have their commensals reach this sort of quasi-stable state. So you layer onto the fact that in this particular 3-year period where all of the action is going on in setting up the microbial community, we also dump the most antibiotics per capita into that population. And so there's studies now out that show that the 0 to 5-year age range, in the US at least, and this is probably true globally, those kids get more antibiotics per capita than any other equivalent 5-year bin. And so then with that in mind, with this sort of vulnerable ecosystem with massive perturbation, we're trying to understand what are the factors that influence the establishment and dynamics of the resistome, obvious factors like antibiotic use and abuse, and then also other correlates which might be particularly impactful, such as disease states and interactions with the environment. And so a pilot study that actually just comes out in press today that we applied these methods to, this functional metagenomics methods to, was to a group of 22 kids with an age range of 0 to 3 months all the way up to 19 years of age. Fecal libraries were created from each of these kids, and then using, again, functional metagenomics, we screened them against the panel of 18 important clinical drugs. And essentially, don't worry about all of the colors here, but what you mostly want to observe from this plot is that anywhere you see a color is where we are able to clone out and sequence an antibiotic resistance gene. Anywhere that's white, we're not able to do so. And this is a much more colored than white plot. And also qualitatively, I hope you observe that the kids who are in the youngest bins, the 0 to 3 months or 0 to 6 months, have about the same amount of rough antibiotic resistance as do the really older kids. And this is one of the major findings that which we're going to now continue to look at, which is that the resistome appears to be established extremely early in life, and at least phenotypically doesn't seem to be that different from later years. And so this is a bit of a busy slide, but just to emphasize that with this very pilot study, just from these 20 or so healthy kids, we were able to specifically identify over 2,500 functional antibiotic resistance genes. And to put that in context, that's a little over 30% of an increase over all known resistance genes in the best database available at this time, the ARDB. And so this method really does allow you to get deep into this extremely critical clinical genotype. And so these are all of the genes that you find over a variety of different antibiotic categories. And then one of the most important things that I want to emphasize is that the reason we spend so much time on the computational side trying to reassemble all of these genes into the constituent context is because we understand something about context. So it's not just the resistance gene, but what's around it. And so, for instance, this shows you a distribution in each plot versus each antibiotic for the number of genes that are found only on their own as an antibiotic resistance genes, the ones that are found synthetic or right next to other antibiotic resistance genes, and then in green, those ones that are found both in a multi-drug resistant context, but also next to mobilization elements. And so essentially by binning this, what you're doing is for each antibiotic class and each antibiotic compound, you're doing a sort of risk factor analysis. You're saying if they're single genes, maybe they're not so critical just yet. If they're multi-drug resistant clusters, now this is the problem of multi-drug resistance moving around. And then the worst problem is if they're right next to genes that already have the ability to pop over into other organisms. because that's the greatest risk for transfer. And unfortunately, you'll notice the majority of these antibiotic resistance genes have at least some examples of these multi-drug resistance clusters next to mobilization. And so I'll leave that. We've got a lot more studies. I'm happy to talk to folks offline about the human microbiota, but I'll transition over to a habitat that sometimes people don't think of immediately when we think of antibiotic resistance, and that's the microbes associated with the soil. And so why look at the soil microbiota for potential exchange with pathogens? But as it turns out, there are decades worth of studies that show that the soil resistome is actually quite important. We know from studies that it's ancient and it's diverse. And the main reason for all of these studies in terms of intuition as to why we would find this is because most antimicrobials that we use in the clinic are actually natural products of soil microbes. So the actinomycetes in the soil have been producing antibiotics for millions to billions of years. And so they've provided all of the selection pressure required, at least over evolutionary timescales, to have resistance. And then we also know from studies where you have archival soils, where resistance levels of known genes have been increasing ever since we came on the scene. And so our hypothesis then was to look for a specific fraction of the bugs that you might find in the soil, specifically multi-drug resistant organisms, and study their ability to have exchange antibiotic resistance over recent timescales with pathogens, again applying functional metagenomics. And so we did that. Fortunately, we were able to repurpose an earlier study where we had isolated such bacteria that were extremely drug resistant. The details are already published. But the point was we were able to go to 11 different US soils and isolate a large number of cultures that were resistant to extremely high concentrations of different antibiotics, and then treat the pool of these cultured sources as a sort of a mock metagenome. And then again apply the same idea of this functional cloning selection and computational annotation to look at the resistance genes in this particular community to try to answer that question of exchange. And again, there's a lot of details, but I'll emphasize the major point that this study showed, which was that we were actually able to identify about 7 or 8 antibiotic resistance genes in these non-pathogenic soil bacteria that were exactly the same, down to the nucleotide level at 100%, to major antibiotic-resistant pathogens. And so collectively, these resistance genes kill 5 different antibiotic classes. They represent all of the known major resistance mechanisms. The pathogens that they hit include gram-positive and gram-negative bugs. And then this is perhaps the telling plot here, which shows a non-exhaustive list of countries shaded in darker colors where pathogens were deposited, which have exactly the same resistance genes as these non-pathogenic soil isolates from the US. And so really emphasizing that this is a global problem. And so with that, I'll end with emphasizing that the reason we think it's important to develop these methods is because resistance selection and exchange is actually an intricate web. And what we'd really like to do is, by increasing the throughput and quantitation behind these methods, is essentially on each one of these predicted interactions, put rate constants and probabilities to say, here are the areas that we really should be focusing on when we try to curb this increased transfer of antibiotic resistance. And so with that, again, I'll thank you for your time. Happy to answer questions later on. And of course, thank the folks that are in the lab and that we work with, our collaborators externally and internally. And of course, none of this is possible without money. So thanks.
Wow. Well, thank you, Dr. Gautam Dantas, and thank you again, Dr. Jack Gilbert, for your first presentation. So we'll move now to Dr. Lita Proctor and her presentation, and then at the end, we will open it up for questions. So we encourage you all to go in and put into the chat box your questions. And at the end, there will also be a survey asking people what else you would like to learn in future webinars. And Dr. Proctor, if you can just go up to the top where there's an arrow and move the arrow to the next page, I think you'll get the slide for your presentation.
Got it. I've got it. Thanks a lot. And you can hear me fine, right?
Yep.
Hello?
Great. Thanks. Yep.
Can you hear me?
Yes, we can hear you.
Great. All right. Thanks a lot. All right. Thanks, Lois. Great. Thanks a lot. That was fabulous, Jack, and that was fabulous, Gautam. I'm unlike Jack and Gautam. I'm not a practicing bench scientist. However, I do have the great privilege of overseeing what I would call sort of the mother of all microbiome projects, and that's the Human Microbiome Project. I would call a foundation project because it did kickstart a lot of fields and also brought to bear the understanding of the role of microbes in the human body that are not pathogens. So I'm a program director. I'm with the National Human Genome Research Institute at the NIH. And I want to tell you a little bit about the NIH Human Microbiome Project. Certainly we've known for centuries that microbes live in and on our bodies. The Dutch draper, haberdasher that had the hobby of making microscopes, the thing he did immediately upon grinding those lenses was to start sampling his own body and everybody else's body. So Antony von Leeuwenhoek's animalcules are really, in my mind, the first real human study of the human microbiota. And just a couple of quick facts, to give you a sense of context. Your human brain as an adult is about 3 pounds in weight, and you're carrying around 3 pounds of microbes in your gut. So it's pretty significant volume and density of microbes. Here's sort of a slightly more biological fact. Something like 60% or half of your stool, dry matter, is microbial mass. And Gautam already mentioned that there's a 10-to-1 ratio of microbial cells to human cells in the whole human system. So the point being, though, that anywhere you look, pretty much anywhere you start looking on the human body, you find microbial cells. And here's just a quick list of direct count values in most cases, whether it's the mouth, the lungs, the skin, the GI tract, and so on. Everywhere you look, you see very significant numbers of in this case, in most cases, these are bacterial cells. There are some data that are starting to come out looking in unexpected sites like breast milk, or in the placenta, where there are estimates of pretty significant microbial cells there as well. So the point being is anywhere you look, you see microbes. So we've known for centuries that we have animalcules, and if you do microscopy, you find bacteria everywhere. Why haven't we actually known much about the human microbiome? Well, certainly as particularly Gautam's presentation showed, we've been quite concerned with infectious disease, and we've developed all kinds of arsenals, weapons to fight infectious disease. In fact, that's been our attitude about microbes, is that they are agents of infectious disease. So we've used vaccines and antibiotics and so on to fight against all of these different types of infectious disease, as I list here. So our main attention has been on thinking of microbes as infectious disease agents. But towards the end of the Human Genome Project, which is now roughly 10 years ago, the microbiology community really called out to NIH that it was really time to embark on a comprehensive genomic inventory of that large portion of cellular life that lived within the human body, the endogenous microflora. In fact, that was even before the term microbiome was coined. So there was a recognition that there was a vast array and diversity of microbes that lived with our bodies that weren't pathogens, that weren't germs, and so it was time to analyze them. And in fact, probably the other key aspect of it, which is really highlighted in both Jack's talk and Gautam's talk, was the ability to apply sequence-based technologies to analyze these populations. As Gautam pointed out, a vast majority of the microbes that we live with are not traditionally culturable, so we missed the large fraction of diversity of microbial life in our bodies. And that really dictated the structure of the first phase of the Human Microbiome Project, which ran from 2007 and just ended last year. And it really was a mapping or the survey of the human. Not only a taxonomic analysis of the microbial community living in and on the human body, but also an analysis of the collective genetic potential in these microbial genomes that known as metagenomics. And this work was done using 2 human cohort studies. HMP, in fact, did not conduct any animal studies. All the work was done on humans. The first cohort study is what we fondly know of as a healthy cohort study, in which 300 adult men and women were clinically verified to be free of overt disease across 5 major body regions. So this is way more than you would get in your annual exam with your doctor. You'd have a dentist, a GI person, a dermatologist, and so on. Each type of clinical specialist would verify that each of these volunteers were free of overt disease. They could not be taking antibiotics. They could not be taking probiotics. They could not be taking immunomodulators. There were all kinds of criteria that were set up to make sure we were really sampling a so-called healthy population. They were tracked over 2 years. 18 body sites across these 5 major body regions were sampled simultaneously, and phylogenetic analysis, the taxonomic data I referred to, as well as metagenomic data was collected on those volunteers. So that was one of the cohort studies. The other cohort study is what we fondly know of as demonstration projects. And they were called demonstration projects because they were really meant to basically evaluate whether there were characteristic microbiomes associated with cohorts of specific diseases. So you'll see that we had sort of 3 kinds of body regions that were zeroed in on. There were a couple of skin disease cohorts. There were several GI disease cohorts. And there was a couple of urogenital, both men and women, disease cohorts that were investigated. And the goal there being not looking at cause and effect, but really trying to evaluate whether there were characteristic microbial communities associated with individuals with particular diseases. That was sort of the first step towards trying to evaluate cause and effect. Just to give you a big picture view, this was quite a large undertaking. There were, well, you can see I've listed them all over the country, sequencing centers and demonstration projects and clinical labs and so on. This entire activity was undertaken as a research network consortium. There were 300+ scientists across 80 different institutions involved in the collection and analysis of these data. And here I want to just tell you that the NIH definition of human microbiome is the full complement of microbes living in and on the human body and their collective genes and genomes. Sometimes you'll see different definitions. So I'm not going to go through all the results that came out of this first phase, but I do want to call out one issue that led to our discussion about a phase 2. So one thing that came out of the study of those healthy cohorts was that each of us are host to about 1,000 bacterial species in our genomes, but that in fact the HMP predicted that for microbiomes in healthy U.S. adults, there may be a global pool of upwards of 10,000 bacterial species and upwards of about 8 million unique bacterial genes, just the bacteria. Remember, when we talk about the microbiome, we're referring to viruses, fungi, phage, bacteria, archaea, but in this case, we're just zeroing in on the bacteria. So if each of us is host to around 1,000 species, that means in any one room, you're going to have a much larger pool than 1,000 species. In healthy U.S. adults, you may have 10,000 bacterial species and 8 million unique microbial genes. So it's quite clear then that the genetic potential for the microbiome is tremendous. And in fact, if you think about those 8 million genes of the human metagenome, that's really considered the genetic blueprint. So we really want to know the next step in that. What's being expressed out of that blueprint? What's the active fraction, the biological properties of the human microbiome? Well, it would be impossible and daunting to even begin to tackle an analysis of all those 8 million unique microbial genes. The question becomes, what are the properties would we need to understand, we need to collect in order to understand the microbiome role in health and disease? Would we only need to know gene expression, the transcripts? Would we need all the enzymatic activity? Do we need to go whole hog and collect all the metabolites, all the metabolic byproducts that are made by the microbiome? Well, no, we can't do all that. So instead, we designed a smaller phase 2, just got started this year, to create a data set of these biological properties of the microbiome and the host. So we didn't do that in HMP1. We really zeroed in on the microbiome, but this time we decided in phase 2 to really start collecting different data types, not only from the microbiome, but also from the host. So from the microbiome, obviously composition, we definitely need phylogenetic data and metagenomic data, but we want to start collecting metatranscriptomic data, metaproteomic data, and metabolomic data from the microbiome, as well as those equivalent properties from the host. And that this combined data set of microbiome and host properties would be the product of this second phase of HMP. The goal being, and I just shot up these 3 data sets, we ran the RFA, we were making awards to 3 projects, one on IBD, one on diabetes, and one on preterm birth. The goal being here is that these were 3 exemplar models of microbiome-associated conditions, and that the combined data set produced from these cohorts, an IBD cohort, a diabetes cohort, and a preterm birth cohort, would help the larger scientific community really evaluate what other properties of the microbiome and host, particularly the microbiome, are going to be essential to understanding the role of the microbiome in disease and health. So we're trying to kind of give the larger community a little help by producing this very kind of pricey and carefully vetted data set so they can go into these data sets and evaluate which properties, combination of properties, appear to be the most valuable and going to that next step to look at cause and effect. Unlike phase 1, you can see that the U.S. map on phase 2 is far less populated. There are 3 research teams. One team is at a combination of Mass General and Harvard. The second is a combination of Stanford and WashU, and the third team is at Virginia Commonwealth University. Much smaller research consortium. However, I think in some regards, even the complexity and the difficulty of what they're supposed to produce is perhaps even higher than in the first phase. In the phase 1 of HMP, the consortium produced 2 important flagship papers that came out last year. We negotiated with Nature to make sure they were open access, so the framework paper and the structure, function, diversity paper, the DOI numbers are listed there. As well, we published about 20 or 25 what we call companion papers, which are studies that drill down deeper into particular data sets or the use of particular tools or analyses, and that's in a PLOS human microbiome collection, and the DOI number for that is there as well. So I don't remember why I decided to include this. Oh, I know why I decided to include this. The other point I want to make for you to kind of start to wrap up my talk is when HMP started in 2007, so let me tell you that in the NIH, National Institutes of Health, that's plural, there are 27 institutes and centers. And back in 2007, there were maybe 3 or 4 different institutes that were doing some work in the microbiome arena. Last year, when we did that same kind of survey, there are now 16 institutes and centers across the NIH that are supporting microbiome-related research. So the interest across the NIH has really expanded. And part of the reason it's clear is there is this list of potential microbiome-associated disease that's growing every year. Almost every part of the human body, you can see a putative role of the microbiome, putative, mind you, role of the microbiome in these diseases, everything from skin diseases to cardiovascular diseases and so on. The last set I listed is mental health condition disorders and diseases, so you'll see this list growing. Now, as I mentioned, the NIH consists of 27 institutes and centers. In 2006, they were funding microbiome-related research at a rate of around $7 million a year. Over the period of the HMP era, HMP alone has been funded at a level of $195 million over 8 years. And outside of HMP, because I want you to compare and contrast these numbers, the other institutes are funding microbiome-related research at almost an equivalent level as the total amount funded for HMP. So in other words, a very healthy level of support for, and that will only continue to grow within the NIH. And these black circles around the institute show the 16 different institutes that are really taking on the microbiome in a big way in their own institutes. So to wrap up, the best place to explore the HMP Phase 1 data tools and protocols will be to go to our Data Analysis and Coordination Center, the DAC, the HMP DAC, and the link is listed above. And they've done quite a lovely job having people look at the tools that were developed for the analysis of the metagenomic and taxonomic data for the HMP, as well as the various pipelines and the various derived data sets that came out of the HMP study. If you want to learn in general about the HMP, not only Phase 1 but Phase 2, I recommend you go to this link, which is to the Common Fund office. The Common Fund is the office under the director of the NIH that funded the HMP, and they have quite a rich website about what we're doing in the HMP, both Phase 1 and Phase 2. We also keep a running list of all the papers that are coming out of HMP support. We're at almost 400 papers, and that that list every year continues to increase almost logarithmically, and I believe I'm done.
Great. Well, thank you so much, Lita. We really appreciate your presentation. That was Dr. Lita Proctor. And we're going to now turn it over to all of you to send in your questions, and I will then ask the questions of our panelists today. So I'm going to jump in with a question for Dr. Jack Gilbert. The first question here is, can the model that you showed earlier account for things like horizontal gene transfer allowing an unexpected taxa to bloom?
No. No, we rely on Gautam to help us with that. Well, essentially what we're looking at is we're looking at higher level community variables. So we really do consider the communities to represent pools of taxonomic groups. So in essence, instead of looking at the response of individual genotypes, which is mired in problems regarding horizontal gene transfer and other elements, it's far easier to consider the community as a unit, an interacting element that has emergent properties that we can actually measure and define. We're working on the more detailed characterization, but we're not there yet.
Okay, great. Well, thank you. And as we go along, if any of the other speakers have something that you want to add, just go ahead and jump in. But I'm going to refer this next question to Dr. Dantas Gautam. Are there observations indicating that multiple resistance can be acquired equally easily by all pathogens? Or are some species able to become resistant or able to acquire certain types of resistances easier?
I think it's definitely the latter, at least in terms of what we've observed so far, both in the clinic and the environment. So there is some suggestion that microbes, and this is now on the taxa phylogenetic level, are particularly specialized at A, horizontal gene transfer, and B, in terms of cross-habitat movement. And so this is something, for instance, that Jack's hospital microbiome project will be particularly good at looking at, and this is why some of the built environment stuff is so critical. So that's sort of the transfer of bugs themselves. But yeah, so for instance, our work has suggested that the proteobacteria, which are a minority of the bugs in the human microbiome, but a majority of one of the largest groups in the soil, there are specific types of proteobacteria that have become sort of professional horizontal gene transfer experts in terms of acquiring and disseminating resistance, and then also have the ability to hop between habitats. So I definitely think that it's not all equal, and there appear to be, badder bugs than others.
Okay, great. Well, thank you very much for that answer. This is perhaps to everyone. With the ever-increasing collection of microbiome data, are there standards in place to enable data comparability and accuracy? Are standards needed?
Well, I can pipe in, although I think Jack and Gautam are actually part of this group. There's certainly the Genome Standards Consortium, which has been in business now for quite a while, which is a grassroots organization that's all about developing data standards for genomic and metagenomic data. Then there's a separate, recently formed grassroots group out of the Human Microbiome Project Research Consortium, primarily, of folks who are actually currently trying to develop data standards in order to be able to compare data across data sets, not only in the U.S. but around the world. And you'll hear a lot more from that group within the next year or two. So yes, there's a lot of active effort to develop common sampling protocols for sampling the human body for the microbiome, extraction protocols, sequencing protocols, and analytical protocols to be able to compare data sets around the world.
Great. Did anyone else want to jump in with an additional piece to answer that one?
Yeah, I can chime in, and even though I think Jack is a better expert on this point, but one of the things that we found to be extremely critical in addition to all of these other protocols is the importance of collecting and disseminating metadata. And so this is something that I think anyone who's interested in getting into metagenome work, I would strongly encourage you to do, and that is take as many data points about what you're going to be analyzing using metagenomics. And you may find that that's even more important than the DNA sequencing itself, because the ability to associate and correlate and hopefully then talk about causative differences is highly dependent on collecting extremely important and accurate metadata about the samples.
Okay, great, thank you. And I'll just encourage everyone to put the questions in the chat box. That would be really helpful. So a question for Jack. What DNA regions did you use to classify bacteria down to species level? The RNA, DNA, or other coding regions from the shotgun data?
It's not very easy thing to do. We use two methods. The first is sometimes the 16S V4 V5 hypervariable region can be used in certain phylogenies to attain a certain degree of confidence for a species identity, but mostly we don't get down below family or genera. So often we end up making assessments of or predictions at the level of family or order. However, using metagenomes and research associates in my lab, such as Kim Handley, we've been applying other algorithms to shotgun metagenomic data to acquire more comprehensive full-length 16S ribosomal RNA fragments, which help us to get a much more effective taxonomic assignation.
Okay, great, thank you. Just to answer a couple questions that are coming through logistically, we will send everyone a recording of this presentation after it's finished. And the other thing is we have posted a survey in the chat box, and we would appreciate people filling out that survey to give us feedback on not just this presentation, but what you'd like to see in future presentations. So here's a question for you, Lita. Who should I talk to learn about the computational approaches used in the analysis of HMP data?
Well, I want to repeat that the HMP DACC, the Data Analysis and Coordination Center, it's hmpdacc.org, has done a fabulous job of including all the computational tools that were used, a whole computational pipeline, the model data sets that were used. I think you'll find everything you could possibly need by going to the website. Certainly Owen White is the director of the HMP DACC, so could answer a lot of questions for you. But I think you'd learn quite a bit by just perusing the website, hmpdacc.org.
Okay, great. Thank you so much. So a question for you, Gautam. You stated that resistomes are acquired early in life. How about for antibiotics that are not administered until later in life?
Sorry, is the question then what happens to resistance genes for which there's no direct selection? I'm assuming that's the level of question. So this sort of takes me to what I think is a critical change for us, and that is thinking about antibiotic resistance as an ecological problem as opposed to a purely microbiological or clinical problem. And that is to say that the selection pressures that are obvious, that is antibiotic use, certainly explains some of antibiotic resistance, but there are many, many other factors, both evolutionary scale ones as well as sort of functional repurposing, which can cause resistance to be there even without selection. So what I mean by that is, first of all, I didn't go into detail here, but antibiotic resistance, I should emphasize, is probably a million-year-old problem, hundreds of millions of years. As long as there have been antibiotics produced in the environment, there have been resistance genes. And so you might have resistance genes popping into a sterile environment like the human gut, simply because they're carried around by bacteria that have evolutionary reasons to carry them around. And then secondly, you have what antibiotics do is they work because they target very, very specific key cellular nodes in a bacterial cell, and it makes sense that bacteria have orthogonal mechanisms to protect those nodes. And so, for instance, bacteria have lots of different pumps to get rid of toxins, and what we find sometimes is that you might have a gene that works as a resistance gene against an antibiotic, but may not be sort of professionally evolved to do so. It's just repurposing a toxin remediation function, which now can also work against antibiotics. And so that's the reason why you might find resistance genes very, very early in what was otherwise a sterile ecosystem, even though they were not drugs right away. And then what happens is when you do apply the drugs, now you give all of the selection pressure for the abundance of those genes to expand.
Okay, great. I know we're a little bit past the hour, but we just have 2 more questions that have come in. And so we want to ask those to the panelists. But there will be a recording sent out afterwards for those of you who are not able to stay behind and listen to the answers to those questions. So the first one, the last 2, again, would be for you, Gautam. So antibiotic treatments are common practice to treat diseases, sometimes even prescribed by doctors just in case. Since emergence of antibiotic-resistant bacteria occurs in the past decades, should we think of alternative ways of treatment and prophylactics? Will we even win the battle against pathogens?
So I'll take that last question first. I think we should never think about it as a battle that we're going to win and be done. Bacteria will evolve, and they'll evolve faster than we do. So I think in the context of resistance, I should emphasize probably the most important thing in this field is indeed to look for new drugs and new therapies. And so one is this idea that we absolutely need to change our antibiotic use practices. This is not only in the clinic, but also something I didn't have time to talk about, but we have lots of projects on this, that antibiotic use globally is highest in the animal agricultural industry. Something like 80 to 90% by weight of all antibiotics in the world are used in animals, not in humans. And so those are practices that warrant serious change. But then also, as the question alluded to, there are all of these other things that we should be considering, which are not just sort of killing compounds against infectious disease, but for instance, there are lots of really neat preliminary trials going on now to look at antivirulence factors. So this is targeting just the aspect of a bug that makes it pathogenic, not necessarily killing the population, which is going to give different types of selection pressures, and that may allow us to sort of, at least in the short term, win the battle or win the first skirmish in a different way.
Great, thank you. So the last question is, someone said that they are hoping to lead microbiome work with high school students. Do any of our panelists today have groups that support outreach projects? So I'll just ask Jack first. Do you have anything?
Yes, we do. Through the Sloan initiatives, we run a number of outreach programs into local schools around San Francisco and Chicago. But there's no reason why we couldn't expand that option. I strongly encourage the asker of the question to contact me, and we can discuss that further.
And we'll put all of your emails into our follow-up of this recording, if that's okay. But Gautam, do you have an answer for that at all?
We don't have anything quite as direct as Jack does, but I would, again, encourage the presenter to get in touch because we have other types of more global outreach for especially high school student. So, for instance, we have a pretty strong graduate student-run program here at WashU for bringing especially disadvantaged and lower income students into science and engineering, and so there are definitely some areas of synergy there. But
great, and for you, Lita?
Well, I can't speak for NIH, but I can speak for the American Society for Microbiology and the American Academy of Microbiology, in which there are outreach programs for high school level students. And I'm sure if they don't already have a microbiome module, they'll definitely have one. They're in the midst of putting together a manual called Frequently Asked Questions on the Microbiome, and I think there's an outreach component that's being developed as well. So I recommend they contact the American Society for Microbiology.
Great, thank you very much. So just a reminder to everyone to complete this survey that's in the chat box, and a great big thank you to all of our speakers today and to everyone online for this fabulous questions, and we look forward to having everyone join us at our next webinars. Again, thank you to Jack, Gautam, and Lita, and everyone have a good day.
Thank you.
Thank you. Take care. Bye-bye.
Bye-bye.